Okay.
Okay,
Yeah.
otherwise I'll keep going. And then
Okay.
we will be able to give you a link.
Yeah.
After that. Now, you should be able to access that yourself. If you have any problems, email us. We'll get it for you.
I'll get it for you and email it to you.
Oh, beautiful. Thank you so much. I really appreciate it.
Okay, we're set to go.
We are live after those technical issues are handled. Good. So folks want to learn fMRI. What primary disciplines are you coming from? Is it many social psychologists and cultural psychologists, which I take as a branch of social psychology. Any other backgrounds?
A couple of anthropology.
Anthropology. Okay. Good. So that's good to know. And then people, many people have taken like statistics. Good. All right. So that's the kind of, yeah. Yeah, of course. Yeah. That's all we need. Yeah. And of course, multivariate optimization in the lambda calculus. Okay, good. All right. That's a joke. All right. So fMRI analysis, the big picture. So the goal here is to do an overview of everything, and then later on, perhaps not me, others will fill in the individual steps. And you guys can interrupt throughout and ask questions, and hopefully this grain where we just do the big picture, it should be readily understandable. If it's not, then stop me. So one thing is, here's the scanner. You put somebody in it, and their brain is doing stuff. Their neurons are firing. Blood flow is altered. And the BOLD signal, the blood oxygenation level dependent signal, is varying in accordance with the neurons firing. That in a way, that's all you need to know about the nuts and bolts of the scanner and the mechanisms by which it operates. More neuronal firing equals more BOLD signal. Now, there is a deeper story to be told. Why is it that more neuronal firing equals more BOLD signal? To be honest, I don't know the details that well. So that just shows you that you can kind of black box this issue for the most part. But here's the molecule hemoglobin. And when you have more neuronal activity, there is more blood flow to regions, there's more blood oxygenation, and there's more fMRI signal. Okay, so if you want to know the intermediate steps, it looks something like that. And here's something that should look a little bit perplexing. Why is that arrow going that direction? I mean, you would think that you have more neuronal activity, it would consume a bunch of oxygen because the neurons are eating up oxygen. So why is blood oxygenation going up? Does anyone have an answer to that? Yeah, go ahead. Overcompensation, exactly. It's to put it in psychological terms, the vasculature overcompensates for the consumption of oxygen locally, and it brings much more blood than the actual neuronal activity warrants, and that's why the blood oxygenation is going up. But ultimately, this is the real thing that needs to be keeping track of. More neuronal activity, more BOLD. Okay, so far so good. So some terminology in fMRI. When we put somebody in the scanner, we collect a high-resolution image, and you can just kind of look at it and see that's a beautiful anatomical image with lots of detail in there. And then we collect a lower spatial resolution time series image. So this is like the movie of the brain that we take over time, and we call that a T2 image, and this one is a T1 image. I'm going to be using this example throughout. We put somebody in the scanner, we take a movie of their brain, and then we put them in different kinds of conditions that we manipulate. And the one that I'll be using is just faces, houses. Repeatedly, I'll be talking about phases and houses as if I'm obsessed with the issue of how the brain processes faces versus houses. So let's go through the major phases of the fMRI analysis. The first one is pre-processing. And the picture you can have in your head is that on the left side, we start out with subjects. Each one, we've taken a high-res and we've taken the low-res movie of the brain. And each one, these movies are collected in slightly different ways. The subject's head orientation is slightly different. There's various artifacts remaining in the data. And after pre-processing, look how nice and pretty they are. Everything is organized. Everything is in the same space. Everything is co-registered. And we're ready to do subsequent analysis. So there's a lot of intermediate steps that go on in pre-processing. But ultimately, what it is taking the movies and taking the high-res and getting them all in the same space. So that's pre-processing. The next step is first level. And here, the movies and the high-res are all in the same space. And what we do is for each subject, we compute an activation map. How we do that, I'll say more about the modeling steps that we do to generate that. So for each subject, there's an activation map. And so notice there's a big data reduction that's happening here. So if the movie of the brain is 200, 500, 1,000 frames, we, after the first level analysis, for each kind of contrast we're interested in, and I'll say what that is a bit later, we end up with one activation map. So we've gone, let's say, from 500 frames to just one after the first level. Per subject. That is, we have one per subject. So if you have n subjects, we're going to get n activation maps at the end of first level. And then, so that's that terminology, pre-processing, then there's first level, and now there's second level. And this is, some people call this the random effects step. So each subject comes in with an activation map, and then we end up with a group image. So, I might have subjects in one group and another group of subjects in another group, and I do a subtraction. So I end up with a subtraction image. I might do a one-sample t-test. So there's different kinds of group images I can generate. But at the second level, ultimately what we're doing is we're taking activation maps, one per subject, and coming up with one group summary map that represents all our information aggregated together. So we start with 500 per subject, go to one per subject at the end of first level, go to one image at the end of second level. Any questions about that? That's clear? Okay. Yeah.
Are we going to have access to these slides?
Yeah. Yeah, maybe I'll put the PowerPoint, or I could put the PDFs up. Yeah. Ariana.
Is this the type of work that, say, one person would do, or is this more of a...
Yeah, it's a good question. Yeah, one person could do this. When you have a lab, you have kind of specialization, and people will do different parts. Mike Angstad is here, and so he's the brains and the operational manager of our fMRI stream over in psychiatry. And so many labs, to be honest, the researcher may pose questions, set up a design, and then say, Mike, help me out here. And Mike would have written all the elaborate software and code that implements all this stuff in a high throughput way, and then we'll give you your results. And then you'll say, Mike, that doesn't look quite right. Can you run this other contrast? And he would do that. So yeah, the fact is that in many labs, it's not actually the investigator that's doing all these steps. But you can't interact with somebody like Mike unless you know the terminology. And so, yeah, part of the goal here is to give a kind of a high-level view of it so that you could productively interact with people that may implement the analysis. That being said, in many, many labs, it is the graduate students or the junior investigators that are actually implementing all this stuff. There is no other layer of folks that you're interacting with. In fact, that's probably the more common thing. So, yeah, one person would have to master all this stuff. Yeah? Yes, exactly. And that will be covered. That's coming up. That's a step called normalization. It's one of the pre-processing steps. So we'll be coming to that. Yeah, great question. Yeah.
Will you please go back to the previous?
Yeah.
So for each individual, each subject, the way you come up with this picture is the same way you come up with the group picture? Because these are
No.
together. It's the same method.
No, it's not the same. Well, in some sense it is. It's an application of the general linear model in both, but it's different applications of it. And so I should be clear that this is just the overview of the 3 big steps, and now we will spend the rest of the time going through the 3 big steps.
Just keep taking pictures of the brain activity, right?
Yeah.
In this way, just keep taking pictures.
Well, I don't think that's necessarily going to be a helpful way to think about it. But we will go through the steps that get you from the pre-processing steps, the first level steps, and the second level. And then we can see whether that's the right kind of way to think about it. Yeah. Okay. So the general linear model. Yes?
I just wanted to, in the data reduction that you're doing, you're still taking a picture of all areas of the brain that are lit up, and those areas that are lit up there show all activity, or they show enhanced activity?
Yeah, that will become clear as we go through the steps. Yeah, that's not an easy, that's a mouthful to explain what exactly, when I say data reduction, what exactly is being reduced and what's being retained. Yeah. So here's the general linear model. And so statistics can be understood on many levels. There's no doubt about that. And one simplified level is this idea of the general linear model, that ultimately what we're doing is we're fitting lines to the data when we do all these procedures. And it's a very flexible framework, and I'll say more about the GLM coming up. But one of the nice things about it is all these methods can be viewed as instances of the GLM, which means that when we're doing statistical analysis on fMRI data and we use a GLM, these are all the kinds of off-the-shelf methods that we have access to. So let's go through what the GLM is. It's really a beautiful idea, this idea that you fit a line to the data. So here's a very, very simple example. We're looking at salary and years of education, and salary is going up with years of education. For many people in the room, this is a great thing because you guys will be doing many, many, many years of education, and obviously your salaries will be rising in a linear way with that. So this idea is intuitive, that we take data points and we fit a line to it. Well, what's so good about, and here's the equation. The y is the observed values, and betas represent the coefficient on this line. X is your data, beta 0 is the intercept, and here is a mean zero error term. So what is the best fitting line? I mean, many lines could be drawn. Here's one line that I drew where the beta 0 is set way up here. The slope is set like that. And this line is not attractive for many reasons. The deviations of the actual points from this line are quite large, these deviations being represented by red. And so our goal is actually to find the values of the betas that minimize the deviations of the observed values from the line. So we want to minimize that function. And then here's a much better line. And intuitively, we can see why that line is a better line. It's a better fit to the data. And what we've done is we've minimized that in order to find that very nice, attractive line. And ultimately, that's the idea of fitting a line to the data. And that idea can be used again and again and again in more complicated ways. So if that simple idea is clear, fitting a line to the data, then much else falls into place. So this is the simple model of salary rising with years of education. I can make a more complicated model. Salary now depends on education, experience, gender, ability. There's an intercept here. There's still that mean zero Gaussian noise that's there. And notice here that gender behaves slightly different than some of these other things. We might measure these other things continuously, but gender we'll set with an indicator function, taking a value of 1 if the person is male and 0 otherwise. And then that idea becomes important because in fMRI analysis we'll be using these indicator variables. So this idea, I hope many people feel fairly comfortable with it. Even if the intricacies of how the model fitting works, this is the bread and butter of what we do in all our disciplines. So I think hopefully people are comfortable with this. This same idea can be imported to fMRI. So here I put somebody in the scanner. I give them instances of faces, and I'm manipulating this. Then I give them pictures of houses. I go back to faces. I go back to houses. How can I use this same idea of fitting a line to the data to extract the activations in response to these stimuli? That's the question that we'll take up coming up. One more thing I should say from the outset is that we do what's called a mass univariate analysis. So what that means is that this issue of fitting a general linear model to the data is done separately for every single voxel of the brain. I haven't said the word voxel yet, so I should say a little bit about it. When we take that movie of the brain, we have the smallest element that we can resolve or distinguish in terms of its trajectory of bold signal over time. That's called a voxel. So we can take this entire picture of the brain or this time series and break it into individual time series corresponding to each individual voxel. For each of these time series, we're going to be fitting a separate general linear model representing activation in houses versus activation in faces. Okay? So let's go back to this idea. We know this, broadly speaking, how multiple regression works and general linear models more broadly. The very same thing is being done when we set up an fMRI analysis. So here's the bold signal over time. Here is faces. So I have an indicator variable here representing when the subject experienced the condition of faces. I have an indicator variable here specifying when the subject experienced houses, and I'm going to estimate these betas that optimize or minimize the least squared deviation from the best fitting line. That's the basic overall picture of how we exploit the general linear model to do analysis of the bold time series. Now, one thing that, and here I've already said this, that is we're using indicator functions for when faces is on and when houses is on. One thing that should be a little bit different is, this was a continuous variable, salary. The bold time series is a different kind of entity altogether. It's a time series. It's something that's varying over time. Well, we don't have to get so hung up on that. The machinery of the general linear model can still be used to fit time series data like this, and we'll say more about how that happens. But right now, the broad outlines of how a statistical analysis of fMRI data should be clear to people. We'll use the general linear model. We'll predict the bold time series at each voxel. So I'm not going to just be doing this general type of thing just once. If there are 50,000 voxels, if the smallest resolvable unit in my movie of the brain, there's 50,000 of them, then I'm going to fit 50,000 of these models. And for each one, I'm going to specify where the faces occur, where the houses occur, and estimate these betas. Is that broad idea clear to people? How that's going to work? Okay. Yeah. Yeah.
Houses does not have something in common to produce a unique portion of both.
Yeah. Let's see. Is the notion of interaction... see, one of the things here is that the manipulation isn't factorial, at least the way that I've described it so far. So it's not even clear if the notion of interaction is defined here. I'm only giving faces, I'm only giving houses. But you're absolutely right that there could be manipulations where the manipulations overlap in certain ways and in certain contexts, in which case you would want a more complicated model here, one that has interaction terms. And that's a very advanced kind of idea, but it can be done. And it's I shouldn't say it's that advanced. The same machinery that we use here, we would use in that case, and we would just have more complicated regressors that specify the interaction. Yeah. Yeah. Yeah.
Is 50,000 just a random number, or can you give us a sense of how?
Yeah, 50,000 is about appropriate. That's it's all going to depend on what type of scanner you've used and how you've chosen to collect your data. But for common parameters, 50,000 voxels would be very, very common to model. Yeah. Okay. So let's continue with how this kind of modeling works. So here's a little bit more on how the indicator function actually kind of looks. So once again, we're predicting the bold at 1 voxel. Here, I'm saying, how is that indicator function going to look? Well, it's going to be something that turns on when the subject experiences faces and is otherwise off. Similar type of thing for houses. And there's a beta associated with each indicator function. There's an intercept, and ultimately, we're not going to fit the data perfectly. There's going to be some residual. So this is looking more and more like the SPM analysis. When we started with, just an equation in text, and now we're getting a little bit closer to how the actual SPM analysis gets implemented. There's now this issue of convolution. So this indicator function right here, when you turn on a stimulus, the brain's response is not that neurons just come on and fire in this precise shape and then turn off. We actually know that that's not what's called the hemodynamic response. That is, the way that the brain responds to a stimulus doesn't take the shape of this indicator function. In fact, it's known what shape it takes. It takes that type of shape. So what I'm saying is that when we present a stimulus that we know excites neuronal activity, let's say for a duration like that, the response of the brain is going to take that shape. The brain will slowly emit the bold signal. It will actually have a little bit of an undershoot at the start. It will slowly rise, and the order of the rise here is on the order of seconds. So this entire thing from start to midpoint might be 6 seconds. From start to this very end over here, maybe 12 to 15 seconds. So in other words, our stimulus generates a slow, lagging response. So our predictions shouldn't be that the bold will respond just like that. Our prediction should be that this convolved with that is, this mixed in, convolved means mixed in some fancy mathematical way, yields this type of prediction. Okay? What exactly is convolution? Well, that's another idea that can be understood on many levels. And what it is composition of functions. And it's a very versatile way to capture the intuitive idea. And again, all we need is the intuitive idea that when the neuronal response is predicted to look like this in response to our stimuli, then the bold response needs to lag and take a canonical form relative to that. And so we mix the two together to generate this type of prediction. Okay? So those that are mathematically inclined or have an engineering background can understand the notion of convolution in greater depth, but you don't really need to. The basic idea is that our stimulus excites neurons, which generates a slow, lagging hemodynamic response, which is what we predict ultimately. So here, our stimuluses, which used to be indicators over here, are now convolved with the HRF. So the prediction now looks like that instead of that. Okay? And so, in fact, we can actually do away with those little indicator functions and just say that our predictions for x1 look like that, and for x2, that is the thing that's going to be multiplied by the beta, looks like that and looks like that. Okay? Questions about this idea of the indicator function versus the hemodynamic response? Okay, good. So now this is what our model looks like. And it should be clear how we got to this kind of model. We can involve the indicator function with the hemodynamic response. And so let's go with the next step, the betas. So ultimately, what we're doing when we say a beta is large, or when we fit these betas to the data, is we're scaling the size of the predicted bold signal. Okay? So we adjust the beta weights based on the, that is, the shape of this hemodynamic response is going to be adjusted up or down in order to optimally fit the data. So here you can see that right over here, blue is where the face regressor is on. There's more activity to faces in the observed time series. So this beta over here is going to be large, because what the beta is doing is scaling the size of this HRF here. So when there is more activity going on in these little blue bands, then this beta is going to scale up to make a better prediction of the bold. When there's very little activity here in the houses, then this beta will scale down in order to reflect the fact that there's very little activity in the observed bold signal when there's houses. Yeah, the HRF is this guy right here. That is, what we're doing, and so the HRF is the hemodynamic response function. So the indicator function is convolved with the HRF because we want to have a better prediction of what the bold looks like rather than that shaped thing. We want something that's shaped more like that as our prediction. Yeah?
So we determine the indicator function.
Yeah.
Depending on the predictions or the manipulations.
That's right.
So that can change things quite a bit depending on what our manipulations are.
Absolutely. Yeah. These indicator functions reflect your design that you presented to the subject. And, if you had given a very different pattern of faces or many more face stimuli, these indicator functions would reflect exactly what you did. That's right. Yeah. Okay. Yeah.
It sounds like on the next slide when you're talking about how after the analysis, it looks like the size of the activity of the
Uh-huh.
But then you say it can more accurately predict like whether it will be large or small. So is that kind of like a post hoc analysis, so to speak? Like you have to have data before you can actually make those predictions, or?
This is fitting to the data. So I wouldn't say it's post hoc. It's the hoc. It's what you're doing. Yeah. So what you're doing here is you're taking that shaped object and scaling this beta higher or lower to better predict that object. So predict, I could equivalently use the word fit. So in order to fit this to optimally, in order to use this to optimally fit that, I would scale this beta to be large and this beta to be small, and I would generate a prediction or a fit for that, but the fit wouldn't be ideal, and something would be left over. Yeah. Okay. So this is the fitting stage. We've observed the bold signal at a voxel. We know what manipulations we did. We convolved it with the HRF. We fit these betas, we scale these betas to optimally fit or predict that, and we have our residuals. Identical operations went on when we had salary being predicted by age, years of education, and gender. All of those, the betas for gender, age, and those things were scaled up or down to make the best prediction of salary. That nothing different is going on here. It's the exact same fitting a multiple regression model to the data. The only thing different, of course, is we have a time series and indicator functions rather than continuous variables and non-time series. Yeah, so the only difference here is this has the temporal issue with it, whereas the other one didn't. Okay, so now the question is statistical significance. And here, the very same principles apply as in standard multiple regression. So think of the composition of all these things in the following way. So the fMRI signal is our data. That's what we observe. The design matrix specifies what we can explain and how much of it we can explain. And then the residuals are what we cannot explain. So after we've done these operations and scaled the betas optimally, there's some portion of the observed signal that we can explain, and there's a portion that we call the residual that we cannot explain. And the fundamental notion of statistical significance, no matter where you look, is that statistical significance is based on the ratio of explained variance versus unexplained variance. So that, if you're doing an ANOVA, you have the mean squared for treatment over the within subject variance or within group variance or the residual value. A t-test, very similar idea. You can explain the difference between the two groups. So you take the difference between the means and you put it over the standard error, which represents what you can explain. So you can do statistical tests on these individual betas, these over here. You can say whether that beta is statistically significant. And how do you do it? Well, you set up the ratio of the variance accounted for the beta versus the residuals of the model. Okay? That principle of how you determine the statistical significance of a beta, that's not unique to imaging as well. That's exactly what you would do in the salary versus years of education and gender. You would compute the statistical significance of individual betas by doing the variance accounted for by that beta over the residuals. Okay. So far, I think the overall picture is that we're setting up a multiple regression model that accommodates the time series nature of the data for every single voxel, and our conditions are represented by indicator functions. We calculate we scale the magnitude of these betas, and we calculate their statistical significance. Everybody there so far? That's all clear? Okay. Now we'll put a little bit more nuance in the model. Because the way that we were doing it before is we were just saying there's a beta for each condition. And it's a little bit more complicated than that. Because when we actually do an fMRI analysis, people don't just sit in the scanner for one long movie. It turns out that what you need to do is break up that movie into chunks. And there's a couple different reasons to do it. One is that subjects can't sit in the scanner without a break or a breather for too long. Usually it's about 6 to 8 minutes that they can usually tolerate. Then they need a little bit of a break, and then they can return to the task. So that's one reason we break things up into runs. Another reason is that there is slow drift properties going on in the scanner. So it's some sort of very slow artifactual thing that's going on that contaminates the signal. And it's hard to detect and remove that unless you break your experiment into 6 to 8 minute breaks called runs. Okay? So the bottom line is we invariably break our experiments into, the maximum would be about 6 to 8 minutes. We don't do much longer than that, which means that we have actually a more complicated model. Whereas before there were just two conditions, x1 and x2 and two betas, we're now going to do that separately for each run. So that the x1 and x2 are going to be, so let's do the betas. This says the beta for run 1, condition 1, the beta for run 1, condition 2. This says the beta for run 2, condition 1, the beta for run 2, condition 2. And similarly for this one. So whereas before we just had 2 betas because we had 2 conditions, we now have 6 betas, plus of course the intercept, which is an additional beta. We have 6 betas because we basically replicated the experiment 3 times. Okay? So that's one thing that makes things more complicated. Another thing that makes things more complicated is we also assign a beta for each of the run itself. So what we do is we construct this mega model here, and let me walk you through this. This is actually a very, very common way to display the overall general linear model structure for an entire SPM experiment. The way to read this is the time series is going down the column. So time is going down here. You know, we're used to seeing that this time series over here, we're used to seeing that flip that way. But now it's been oriented this way, and there's good reasons to do that. So this is the thing that we're trying to predict. Each individual beta gets its own column. And so what this is saying is that faces were on in run 1 over here. Faces were on in run 1 over here as well. Houses were on in run 1 here and here. Faces were on in run 2 here and here. Houses were on in run 2 here and here. So this entire complicated model, and in particular when these indicator functions are on, are depicted in this kind of column form in a design matrix. And in addition, we model the effect of run. So I said that the scanner has weird artifact that happens at a very slow time scale. Well, one of the ways we remove that artifact, and there's other ways as well, is we actually put in a beta that represents the mean effect of run. So that's why this set of indicator functions, and these guys over here will be convolved with the HRF, because we don't want that to be our predictor. We want something with a nice shape to be our predictor. This is our overall GLM, okay? We have 2 conditions, 3 runs, so we ended up with betas for 2 betas for each run, an intercept, as well as betas for the run itself. Everybody clear how we ended up with all these betas despite the fact that we only had 2 conditions? Okay. All right. 2 conditions, a lot of betas. That's the take-home message here. All right. And again, this is all just doing multiple regression, exactly like salaries being predicted by years of education, gender, and stuff like that. Okay. And here I've pointed actually to where stuff is represented, but I just went over that. Okay, now comes to the crucial issue of contrasts. We already have all these betas. When we're thinking about the issue of contrast, let's do one thing. The beta for the intercept and this term that represents the mean zero noise, we go ahead and get rid of that. So just for convenience, not just for convenience, just to better represent how contrasts work, think of the model now specified like that. We've removed the intercept and we removed the mean zero noise, and we're just looking at the model like that. Okay? A contrast is can be understood, again, on many levels, including a linear algebra level, but I just want to understand it on an intuitive level. Let's say we want to ask this question: is there a statistically significant activation to faces versus baseline? That's a very important question. We would all, you know, in setting up this experiment, we would pose that question to ourselves. That's why we did this experiment. Well, what we do is for each beta that represents faces, we put a 1 on it. And what that says, actually we do something slightly different. Rather than put a 1, because you know the numbers are fairly arbitrary here, we actually put a 1/3. Our goal is to get all the numbers where we would have put a 1 to sum to 1. Why do we do that? Well, it turns out that it scales the betas in a nice way. When we're doing statistical inference, the scaling of the beta becomes an issue, not because it affects the p-values, but later on when we want to interpret the betas in convenient units, it turns out that this is the way to go. That is, rather than just put a 1 on all the betas of interest, you actually want all the betas of interest to sum to 1. So you go ahead and put a 1/3. You put a 0 everywhere else. This expression, that contrast, expresses the following notion: how much variance is explained by this subset of betas? Okay? So when we start asking inferential questions of our general linear model, the mediator by which we do it is by specifying contrasts. And the way that we specify contrasts is we keep track of all the different betas that went into specifying this GLM, and we pose our question: how much variance is explained by this subset of betas, in terms of positive numbers on the betas of interest that will all sum to 1. Okay. Here's a different question. Is there significantly more activation to faces versus houses? That's another question that we would want to ask. I mean, the reason why we put houses there is we don't want to... Lots of different things happen when somebody looks at faces. Their visual processing gets turned on in a fairly generic way. So houses represents our control condition, where visual processing is turned on, but it's turned on to something that is not a face. So now this question, is there more activation to faces versus houses? Will answer a very nice question about what's selective for faces. The way that we would answer this question is similar to what we did before. The initial idea is to put a 1 on all the betas of interest and a negative 1 on all the control condition. And again, the same principle gets followed. All the positive numbers sum to 1, all the negative numbers sum to negative 1. If you do that, later on, when you do more sophisticated analyses called ROI analyses, the resulting betas that you look at in graphical form will be in units of % signal change. So that's a very advanced idea. You know, we're not doing ROI analyses quite yet. We're not looking at % signal change. But this way of scaling, of setting up the contrasts will ensure that the betas have nice scaling properties. Okay. So that's a contrast. And you can, I think, knowing these two things, that is, making comparison against a baseline and making a comparison against two conditions, that's the lion's share of kind of things that we do when we interrogate a first-level model. Okay? So is that clear? No questions about that? Yeah. 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 The runs always get modeled with their own regressors, and yeah, and they're block regressors. Even in event-related design, the runs always take a block kind of shape. What would differ is the indicator functions for faces, houses would take a very different shape, and the resulting predicted bold would take a very different shape after convolution with the HRF in an event versus block design. And that's a good general point. I haven't said so much about event versus block designs, but for those that are familiar with it, the mechanics of the GLM don't change one bit if it's event versus block. The only thing that changes is your indicator function that you specify will take a different shape. But every subsequent step, convolution, setting up the betas, interrogating the model with contrasts, everything happens identically thereafter. Okay. Any questions? Any more questions? Yeah.
So what if you have, say, 3 conditions?
Yeah. Yeah, I mean, it scales up. The same things that, so now I would have beta 1, 1, beta 1, 2, beta 1, 3, beta 2, 1, 2, 2, 2, 3, and yeah, so I would end up with more betas, and my contrast would have to be changed a bit to reflect the questions that I'm posing. So if I have faces, houses, and vegetables, and I'm interested in faces versus houses, I would just make sure to put a 0 on vegetables. Yeah, right. Okay. Again, mass univariate analysis. That is, everything that we've done so far, we do 50,000 times. And it doesn't sound as, it's not as awful because we fit the same model 50,000 times. It's not as if we're fitting different models for different parts of the brain, although you could do that in theory if you wanted to. So ultimately, when you specify a model for the software replicates it 50,000 times for each voxel of the brain. And this is a question that was posed earlier. What is that data reduction actually doing? Well, when I fit a model 50,000 times, I get 50,000 p-values, right? I mean, we know how to get that p-value. It's the ratio of explained variance to unexplained variance. So I get 50,000 p-values, and I threshold the p-value. Very roughly, I threshold the p-value, and then what gets retained is a thresholded map, where everything that didn't meet that threshold is not present here. Everything that met the threshold or higher is then displayed with gradations of how far it exceeded the threshold represented in the coloration of the map. So when you look at a paper in a journal like Science, and they show you one of these maps, it's a threshold imposed on a mass univariate analysis where the threshold is usually something like 0.001. Why is the threshold set so stringently? Because of multiple comparisons. When you do 50,000 statistical tests, you need to adjust for the fact that you've done so many comparisons. So the threshold is adjusted for multiple comparisons. That's actually its own issue, how to handle multiple comparisons in fMRI. But we won't be covering that today. But you can see why there is a need to handle multiple comparisons effectively. Okay, so what that's, well, I should have put another slide here. What we've done, you can imagine, is for every single subject who came in with 500 images, let's say, in their movie of the brain, we came with, for each subject, we now have one heat map. So I should be clear that not only are we replicating this for every voxel, but we're generating one of these for every single subject. We haven't collapsed across subjects yet. Okay? That's where we're headed to next, the second level. Now, each subject comes in with one of these heat maps, and then we create a group image. So somebody asked earlier, is the same kind of mechanics being used when you collapse across time in a first level analysis, and now you're collapsing across subjects in a second level analysis? Yes. Similar GLM type ideas are being used. I'm actually going to go back to the slide before in a second. But here's your first level design matrix where the rows are what? Who can remember? What are the rows? Time. Rows are time. The movie is going downwards. It's starting over here and is finishing over there. Right? Here, the rows are going to be subjects. So this says these subjects are assigned to group 1. That's the white means the indicator variable is on. These subjects are assigned to group 2, and some statistical test is being done. So broadly speaking, yes, the reduction that happens in first level is very similar to the reduction that happens in the second level, in the sense that a general linear model is being fit. But whereas this was more complicated because we have this issue of time, this is much more like a very standard salary equals gender versus years of education or whatever. There's no time element here. It's just a general linear model where the indicator function bins subjects into groups, or maybe you're doing a one-sample t-test, in which case you wouldn't bin them into groups. You would just collapse all your subjects into a single beta. Okay? So GLM is being used at the second level. Now, there is this issue of random effects. In fMRI analyses, maybe before the year 2000, everybody did fixed effects analyses. These days, everybody does random effects analyses. The difference, for your own purposes, you don't have to worry about it because the software will calculate it. But what ultimately is it? Here's a good kind of idea to keep it straight in your head. Here I've given monoclonal I've given fruit flies that are all genetically identical. I give them, half of them a drug and the other half get a placebo. And I want to model the effective drug, so I do exactly what I would usually do. I have a beta representing the effective drug, an intercept, and I have measurement error. The only source of error in this type of model is measurement error, or broadly speaking, things like measurement error, because all of my subjects are identical. Here, I actually, here's human beings, and everybody's different. You got this short, squat guy, you got this tall guy over here. Everybody's different, and my groups are going to have some variation in subjects. Again, I give a drug and I give placebo. I model the effective drug. I have an intercept. I have my measurement error, and now I have one more error term that's different. This type of error is mean zero. This type of error is mean zero. So I have two mean zero error terms. The reason to keep them separate is their properties are different. One error term reflects the measurement error that whatever dependent variable I have, I'm measuring it imperfectly. But I also have this kind of mean zero subject error. Every subject is a random draw from a population of subjects that has certain properties. And so the mean is zero, and then there's a deviation from that mean that reflects the type of subject that I have. Anytime you want to model these two separate sources of mean zero error like this, you call it a random effects model, and its mathematics becomes more hard to handle. And in these models, you're usually going to do some sort of maximum likelihood estimation rather than just kind of ordinary least squared. And so the software does that behind the scenes, and you don't have to worry about it. But that's what that term random effects means. Okay? All right. So to review, pre-processing took us from a movie to another movie, where this movie is all, let's see, actually, this wasn't a review at all. Now I'm going to do pre-processing. Yeah, and then we'll take a little bit of break, and then we'll go to the end. So, so far, what I've talked about is first level and second level. All that presupposed the data was pre-processed in the right sort of way. And I wanted to punt on the issue of pre-processing because in many ways the heart and soul in fMRI analysis is first level and second level. But pre-processing has to be done right in order to make all that rest of it possible. Okay? So, somebody asked, how can we take subjects that, everybody's head is different, the scanner was oriented differently, and so forth. How can we go ahead and enter these guys into a first level analysis? Well, the answer is that we don't enter these guys into a first level analysis. We tidy up the data first. So how do we tidy up the data? There are 4 big steps in pre-processing: slice timing, realignment, normalization, and smoothing. And I'll very quickly go through them. And then, you guys are going to have labs with your GSIs here. Steve, Stephanie, I don't know the fourth. Sarah, yeah. And so they're going to be going through all this stuff with you so you get some hands-on experience. But here are the big 4. So slice timing. What is that about? Well, fMRI data is acquired a slice at a time, and usually from the bottom up. So, here's an egg. I'm taking a slice at a time. Here's a brain. I'm taking a slice at a time. Why is that so important? Well, look at, here's slices of the brain. And here's a voxel at slice 1. Here's a voxel at slice 2, voxel at slice 3. These voxels, the ground truth I'm just stipulating, are identical in terms of their time series. Look at these waves. The waves are absolutely identical. But any attempt to sample what the bulb is doing is going to require some non-zero amount of time, right? So I sample over here on this voxel, and I make this estimate for the bulb. I move up, and a little bit of time has elapsed, so time is going that way. And now the next sample is taken right there. A little bit of time elapses, and the next sample is taken right there, and so on. Notice that despite the fact that these voxels, the ground truth is that they're behaving identically, I'm going to say the bulb is different in these voxels at that time. Why? Because it took a non-zero amount of time to do the sampling in ascending slices. So if we had a amazing, godlike scanner machine that took no time at all, this problem would be alleviated. I would sample right there, there. And I would know, wow, this voxel is doing exactly the same thing across these voxels are doing exactly the same thing across these slices. Well, we don't have a godlike scanning machine like that, but what we can do is interpolate. So given the properties that we observe across time, we can make a best estimate of where this guy would have been if I had sampled him right here, and so on. Obviously, the interpolation is going to apply more correction to higher slices because more time would have elapsed by the time I get to higher slices. Yeah?
You said, I think typically you get one slice every 2 seconds.
No, you get one frame every 2 seconds. So it's one entire picture of the brain every 2 seconds. Slices would go much, much quicker than that. Yeah, right. So that's slice timing. And, the mathematics of the interpolation and all that probably don't need to know. But what this operation does is it's going to interpolate across slices like this. The next step is realignment. So what we do is when somebody is in the scanner and their head is situated at time t0 at a particular place, they may move across time in that same session, in that same scanning session. So what realignment does is it takes the t0 image as the source image, the stable image, and it moves every subsequent frame to be oriented in the exact same orientation as the first image. So what you're doing is if at t0 my head is here, and at t1 and t2 and t3 I'm moving in various places, the mathematical operations here will reorient all of the T1, T2, T3 images to be exactly in the same orientation as T0. It's a rigid body transformation, so you're calculating parameters that, assuming that the head is a rigid body and it ought, you know, the head should not be squishy and moving, it's changing its shape over time. If you assume rigid body, you can actually calculate the amount of rotation and the amount of displacement XYZ and rotation rho pitch yaw that would move the head optimally to be aligned with that first image. So realignment will literally move each frame of the movie to be aligned with the first frame, and will also write out the 6 displacement parameters, XYZ, rho pitch yaw, that were required to move each subsequent frame to the first frame. And those will be written out in a file separately. So not only is the software going to realign things for you, it's going to tell you how much it had to move things on a frame-by-frame basis to get that particular frame to be aligned with the first frame. Okay? Yeah?
Also, I was thinking, like, if you do long sessions, you put the participant back and their head will not be exactly in the same space as the last session. Right?
Yeah.
The last session, or do you just care about that session?
We realign on a, so I think you're using session to mean similar to something as run. Yeah. So remember that each experiment, we usually break up into 6-minute runs, and the subject gets a little bit of a break. Yeah, we realign on a per-run basis. We realign to, is that our usual practice, on a per-run?
So it's actually a two-step. It realigns first to the first image of each run.
Yeah.
And then it realigns each run to the first image of the first run. So that they're all eventually realigned to that first image.
Yeah. So if you ask more in-depth questions, you'll be finding that I look that way. Right. And so Mike is going to be a great resource if you guys have more in-depth questions about this. Yeah, so it's a 2-step process. And yeah, so it's all realigned to the first image of the first run eventually. Yeah. Normalization. Look, everybody's head is different. This is the point that was made earlier. But there are mathematical operations that basically can bend and shift a person's brain to match a canonical brain. And so here's a canonical template. So here's my individual subjects. And there's a canonical brain that has been calculated by averaging a bunch of people's brains, 150 people's brains together. That canonical template, called the MNI template, that's available to us. And so what we do is we take each subject and we calculate a deformation field that will optimally make this guy's individual head most resemble the MNI template. We save these deformation parameters and then apply these deformation parameters to the movie. So the upshot of what normalization does is it takes each individual subject-specific head shape and gets everybody bended and twisted in the orientation of the canonical template. And those functional images, the movie themselves, are also bended and twisted in a corresponding way. So, you know, why normalize? I mean, this should be obvious because somebody posed the question earlier, how can we even do these analyses if we don't normalize? So the intuition is clear. If everybody is bended, is very unique, if I tried to sum across subjects, note this, first level should be just fine in many respects, even if I don't normalize, because first level is across time within the same subject. The problem occurs when I try to aggregate across subjects. Notice this guy and this guy maybe have similar activation in similar places, but I can't aggregate correctly because each subject's anatomy is so different. The second level won't be accurate. So after normalization, you can accurately make inferences across subjects. Yeah.
Since this is people doing cultural neuroscience,
Yeah.
the obvious question is, so how do you get the canonical, that normative brain against which everyone else is compared? Where do you derive that back?
Yeah.
It's a bunch of people from McGill.
Yeah. Yeah.
Sorry, not Montreal.
Yeah.
I mean, they've actually since updated it. It's like the current Marital template, I think it's like 300 or 450 subjects. But yeah, it's some given sample or something.
Yeah. So, this may be an issue if anatomical head issues systematically vary between populations in a way that more deformation would be required for one population versus another. Yeah, that could definitely be an issue. Where it's an even more serious issue is comparisons between children and adults, because there the head shape is definitely completely different, and much more deformation is going to be required for children versus adults. Nonetheless, our best understanding, because I'm in the department of psychiatry and we do a lot of children kinds of things, the template seems fairly robust to the fact that different types of deformations may be required for different populations. Yeah. Yeah.
Is there a way to account for possible data that's lost by normalizing, if data is lost at all?
Yeah, I mean, the deformation, nobody does it. Yeah, but in theory, this is written out. And so one could look closely and see whether there are, experiments or contexts where this is systematically different, and one should worry that there's a confound in the amount of deformation that's being applied in this context or to this group versus another. Yeah. Okay, last step is smoothing. So, each frame of the movie may look like this originally. When we normalize it, we bend it and twist it. Now it looks to be in the shape of the MNI template. And then what we do is we blur it. Smoothing is an operation where each voxel will start to, will incorporate the level of activation of the voxels around it. And the reason for that blurring is that, our ability to register locations in space is not that great. So, I think of it a little bit as the whole issue of significant digits. You shouldn't be using more significant digits than your measurement process allows. And so we smooth it so that the payoff from smoothing, which kind of justifies the procedure, is if you don't smooth your data and calculate a first or second level activation map, it might look like this, speckled. After you smooth it, you get nice smooth activations, and a lot of that speckling is gone, suggesting that some of the problematic things that are going on here is just because space is not aligned across subjects optimally. And the underlying neuronal processes may differ very small ways across space, across subjects. So by blurring, you get a more stable map. And that's what smoothing is. Yeah, so, this heat map, the underlying beta can be larger in one group or smaller. And so earlier I just ignored that issue that the difference between the group can be in two directions. So the heat map by convention, where group 1 is greater than group 2, we color it orange or shades of orange, where group 2 is greater than group 1. So group 1 is lower than group 2. We color that shades of bluish colors. Yeah. Okay, that was a lot. But, the good thing is that's the whole shebang in a way. Everything that you learn hereafter, most of it should be filling in the details of this overall picture to greater and greater extent. Yeah. 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 Absolutely. Yeah. This is a huge issue that, as people learn more and more about the brain, we're now starting to ask questions not just about small structures like amygdala, but, ventral tegmental area or habenula, tiny, tiny structures. And so For those kinds of questions, when people pose questions, probably the thing to do is optimize the scanning sequence, collect data at finer voxel resolution, using smaller smoothing kernels, maybe even different smoothing kernels for different regions of the brain like you suggested. I don't know an easy way to do that because the software, as far as I know, is going to apply smoothing homogeneously across the brain. But you can change things. It's all written in MATLAB, so you can change things. But as people pose questions about smaller brain regions, it becomes more and more important to keep in mind what some of these things are doing and optimize the protocol accordingly. Okay, so the take-home message is when you put somebody in the scanner, their neurons fire according to your manipulations, and bulb goes up or down accordingly. In pre-processing, we go from a movie, say 500 frames, to a movie, again with 500 frames that's been cleaned up. And first level, the movie of 500 frames per subject, now it is modeled with a GLM, which is structured appropriately for time, and you get activation maps, 1 per subject. In second level, you go from activation maps, 1 per subject. Again, you use a GLM, which structure is not oriented around time, and you get group image. That's the big thing. Yeah, that's the overall flow. Okay. Yeah, now overall questions. Yeah.
For the individual subject activation maps, is it standardized as to have one condition of activation? So like if you're looking at houses and faces, you only have an activation map of faces, or you only have an activation map of houses, or do you ever overlay them?
You can. So if you're interested in visual processing irrespective of content or stimulus type, you would put ones over all the face betas and ones over all the house betas. And then you would, of course, rescale them so they all sum to 1. And I also gave the example of faces minus houses, so that implicates all the different betas, but this time faces get a positive and houses get a negative. So all the betas are available to you to do what you want. The contrast you can set any way you want to. Some contrasts are sensible and some would be crazy. So run on 1 faces and run 2 houses subtracted against each other just doesn't make a whole lot of sense, but you can certainly do that. Yeah. More questions? Yeah.
Sample size.
Yeah.
How many individual subject you make 1 group picture?
Yeah.
Some kind of rule or...
Well, practically speaking, most experiments will have at minimum around 20 subjects. And that's saying what the modal number is. Now, the most common number. The fact is that the appropriate sample size all depends on the power to detect an effect. So if you have pilot data and you know that the effective interest has this X level of detectability, then you can plan appropriately for how many subjects you need. The fact of the matter is most neuroimaging studies are probably underpowered. That's a point that I'll come to a little bit later when we talk about other kinds of analyses. But most neuroimaging studies are probably underpowered. We probably need more than 20 subjects. If you are doing group comparisons, so, let's say I'm interested in face-specific processing. So I do a contrast test of faces versus houses. I've done a subtraction at the first level. If I'm now interested in face-specific processing in Easterners and Westerners, I'm now doing a double subtraction. And it's invariably the case. It's not necessarily from the mathematics of it, but it usually is the case that you're going to lose the detectability of a double contrast is going to be less than each of the single contrasts. So the fact is that we're going to need a lot of subjects for the kinds of experiments that many people are interested in. Similar thing in psychiatry. We can easily get amygdala activation to fear faces. To get the difference in amygdala activation in fear faces in social phobics versus healthy, that's, you're going to need more subjects, many more subjects. Yeah. Mm-hmm.
You might need contrasts as
And first level, yeah. Yeah.
Now, when by the GLM at second level, do you do the same with contrast, or
Yeah.
no,
Yeah.
what significant tests are we doing at that point?
Yeah, I mean, yeah, so this is a very flexible framework, this notion of a GLM. So here, I've just partitioned my subjects into two groups. And so if I put a 1 over here and a negative 1 over here, or 0.5 over here and a negative 0.5 over here, I will, actually a 1 and a negative 1, that would have been correct, because all positives sum to 1, all negatives sum to 1. I will represent the idea of what is the difference between those two groups in the form of a contrast. Now, let's say I have a parametric scale variable. I can put another column that represents the parametric scale variable, and I can get the effect of the scale variable of interest. So GLMs are very, very flexible, and anything that can be expressed in the form of multiple regression, you can express in this kind of design matrix of GLM, both first level and second level. But of course, they'll be structured slightly different because one is representing time and one is not. The second level is not. Yeah. More questions? Yeah.
This is really what you're discussing is the treatment experimental kind of model. But one of the articles that we had to read for today, which I'm trying to look up and I can't put my finger on it, was basically, it seems to me, being a total neophyte to this, talking about what the brain is doing even when it's seemingly in a rest state.
Yeah.
And so you're not discussing any of the sort of spurious brain activity to the experiment here at all. Are you going to talk about that?
Yeah.
Later on?
Yeah. The next two things will go by more quickly. One will be about the default network and connectivity. That's right. And then the one after that will be about pattern recognition methods, which are probably where things are headed in the future. Yeah. Yes.
You were mentioning, I think this, let's say you do like a two-culture comparison.
Mm-hmm.
Let's say play subject from each culture, right?
Mm-hmm.
Would want more variance among the subject in terms of the demographic variables, or less variance from each culture? Exact, say, like age, gender.
Yeah.
You want more variance from each culture. What doesn't matter?
Well, I mean, variance due to age and gender, things that you usually do not want unmodeled. That is, so if I know that I'm interested in the effect of Easterners versus Westerners, and I know that the effect of interest, the thing that I'm measuring, Eric, is also susceptible to age effects and gender effects, I will put new columns in this design matrix that model those effects. But I will be clear in my contrast that I'm not interested in those effects. I will put a 1 over group 1 and a negative 1 over group 2 and zeros everywhere else. And that way, I mean, these statistical tests are always ratio of explained variance over unexplained variance. The denominator is going to get quite large if you're not modeling known sources of variance. So if I understood your question correctly, you basically do want to model all other sources of variance that you can systematically account for.
Basically, when you select a subject, it doesn't matter their demographic variables, because you're going to model them anyway.
Well, there's limits to how much these models can account for. So optimally, you'd be dealing with fruit flies, replicates of each other that were given a different manipulation, like culture, the cultural upbringing. Short of that, you have to model sources of variation. Yeah. Right. So I'm just taking issue with that, your claim that it doesn't matter. It certainly matters. And we do want to use homogeneous populations as much as possible, but where we can't, we model the known sources of variance. As much as possible. Yeah. Yeah, I mean, this becomes a very nuanced question of experimental design. Attempts to get homogeneity in population then can corrupt the population that you're sampling from relative to the question you're asking. So there are all these nuance and trade-off kind of issues. But the fact is, focusing on the issue of analysis, we can model known sources of systematic variation like age and gender.
Basically, it's not a random selecting. You do need to have some standard
the substandard is needed. That's correct. Yeah. Uh-huh.
So I know one of our presenters yesterday was doing age and culture effects of social distancing, young and old, U.S., young and old, Singapore.
Uh-huh.
You have those types of, I guess, subjects where you have one group that you know is young and one group that you know is old, and so you would be maybe controlling less for each subject population because you have a rather homogeneous group, but then you want to compare between them. Can you do those same type of controls between group images, or can you only really do it between individual? Does that make sense?
If I understood correctly, you certainly can make the comparisons that you are suggesting. That this framework of, so I think what you're describing there is something like a 2x2, young, old, and east-west. And that does bring in the issue of interactions. And so the bottom line is that GLM can handle a model like an ANOVA or a 2x2, and it can compute main effects as well as simple effects as well as interaction effects. So one way to put it is anything you can do in SPSS, you can do in the GLM framework. I'll say one very quick caveat. There are known limitations of SPSS in doing ANOVAs, especially within subject. I think Mike was the person that discovered this and came up with the optimal solution for it. We don't have to get into that now, but is this type of analysis tractable in GLM? Yes. Did the people that wrote SPM, did they implement the mathematics of it correctly? No. But there are workarounds. And I will say one quick plug for our methods core that we developed, and Mike is the main person in it. There are efficient ways to do these analyses and some inefficient ways, yes. And so getting the right software stream will really enhance your productivity. Ways in which the data can be organized in ways that facilitate asking scientific questions without you having to muck around too much in the guts of the software or do too much point-and-click operations. And so we've designed, on top of SPM, we've designed a suite of software tools that we think are really efficient for doing the vast majority of analyses that people would want to do. And so we're trying to make those available to groups at other places. So if anybody's interested, let me know. All right, we should probably take a break, maybe for like 10 minutes, and then we can come back and do the rest of the next 2 lectures, which I promise will be less draining on the head and in a way almost more interesting because they get into more interesting questions. Okay, thank you. Yeah. Yeah. Math questions?
Coding, yes. Because, so I was a signals analyst
Yeah.
for 10 years, so I analyze just the straight signals. I have a lot of questions.
Oh, yeah. Don't ask me, yeah.
All right. So you're okay. So I have a lot of questions.
Yep, you bet.
Mustang.
Yeah. Oh, yeah. Yeah, my pleasure. Yeah.
No, I know this because the first time I met
Yeah, I'll definitely be there Monday. Yeah, I'm looking forward to it. Yeah.